We're on the floor of Society for Neuroscience 2022. This is the first in-person SFN since, I believe, 2019. I can't really keep those dates straight. And we are joined by a good friend of MPFI, Dr. Gordon Smith, who's an assistant professor in the Department of Neuroscience at the University of Minnesota Medical School. His research group is part of the Optical Imaging and Brain Science Medical Discovery Team. Gordon, thanks so much for joining us on the podcast.
It's great to be here.
It's great to see you. It's been a while. So you were a postdoc at MPFI. You started your assistant professor position, I want to say 2017.
Yep.
Is that about right?
Yeah, late 2017. Opened the lab in 2018, and then sort of been going since then.
Can you tell us a little bit about what's sort of the major thrust of the research you're doing now?
Yeah, so we're really interested in how large-scale networks get built during early development. So looking in the visual cortex and in other cortical areas, how networks that interact on millimeter scale, how they assemble during early development.
So you have a long history from your PhD work up through your postdoc until now of working in visual system on developmental type projects. What is it about visual neuroscience specifically that you've always gravitated towards? Is it the sort of the tractability of the problems, the types of questions, or why vision specifically?
Yeah, I think it's a system that is tractable. It's a system where you can control the inputs. You can put the same stimulus up in front of an animal at different ages, and you can ask how the system responds to those inputs, how that changes. And then, my questions have sort of recently, or since middle of my postdoc, really shifted to being very developmentally oriented, and I think that's really because trying to understand how the system gets built is essential to understand how the system behaves.
Mm-hmm.
That everything that the system does is built upon foundations that are laid down early in development. So we really need to understand that.
But at the same time, developmental work's really challenging, right? Because there's all these issues of having to time things perfectly with your experiments. What are some of the challenges of working in developing mammalian systems for these types of experiments?
Yeah, there are definitely some technical challenges that we don't always have the length of time to have things express. We have to get experiments to sort of happen faster. We're sort of stuck to certain windows. We also do all our work in ferrets as a model system. They're a brilliant model for development. They have this really sort of unique developmental profile where they do a lot of their development postnatally. So we can study periods of time that are similar to second and third trimester in humans in a postnatal animal, but that also means that there are tools we don't have that are available in mouse that we have to sort of find ways around.
Yeah, actually, now that I'm thinking about it, this only just occurred to me. We've even had David Fitzpatrick on the podcast at one point. I don't think we've ever talked about, the challenges of working in ferrets specifically. So, can you give us a little bit of background on the history of why ferrets are an important developmental model in neuroscience as opposed to, say, mice?
Yeah, so I think part of it is that at least their visual cortex, and we actually think a lot of their other cortices are organized in a way that's very similar to humans. And rodent cortex seems to be very different. And so I think it's a reasonable hypothesis that some of the lessons you learn from mouse might not generalize to humans. And so then if you say, okay, we need a system that models humans in a reasonable way, and also has this window of development, so we can study things that happen really early on, because a lot of our thinking is that a lot of the problems you see, neurodevelopmental disorders that you see in humans once they're born, the underlying cause of those happen much earlier in development. So you need to study these early developmental windows to get a handle on what's happening there and develop therapies.
And I guess, one of the the components of that developmental question is what's the role of experience in shaping the response properties of neurons in the visual system? There's probably a lot of questions you can ask before ferrets, for instance, have any kind of visual input. But one nice thing about ferrets, as I understand it, is that they have this prolonged period where their eyes aren't open, postnatally. So, what are some of the things you can make use of the ferret for, in terms of...
I think that's it, that you have this window of time where the system is doing its sort of normal development, and you can study how the system has changed, how perturbations during that time impact the ultimate development of the system. You can model neurodevelopmental disorders in this system that may produce more realistic or useful models for human disorders later on.
Mm-hmm, mm-hmm. So, coming out of your postdoc work in the Fitzpatrick lab, what were some of the first questions you were interested in either continuing to probe as a PI at University of Minnesota? Like, what were your first priorities basically when you started that new position?
Yeah, I think we were really interested again in this idea of early spontaneous activity, the structure that happens in the brain during early development. And so we were building on experiments that I did as a postdoc and try to get a little bit more mechanistic. We've also had a few additional questions that have sort of broadened out, looked at slightly different areas of the brain, various different projects that we've done over the years.
So with the spontaneous activity, you're using basically functional imaging techniques to visualize, as you said, like broad scale network activity. And you can sort of, what are you able to visualize with that in terms of this network activity? Like if you could sort of paint a picture of what it looks like when you're seeing this under the microscope.
So one of the really powerful things about looking at spontaneous activity is it can tell you what the structure of the network is at time periods when the system can't be driven externally. So we can see that there are regions of the brain that are really highly correlated, that are forming networks that function over millimeters, and this happens much earlier than we thought that they do. We now have some sense of what the mechanisms are that might give rise to this, how those change over time, and how those early patterns of activity shape what the system does later on.
So you basically are saying that because in the absence of external stimuli driving the neural activity of say, visual cortex, the activity that you're visualizing is probably due to intrinsic, intercortical dynamics where regions are responding because of the response properties of other regions, basically.
And it reflects something about the underlying state of the circuitry at that point in time.
Like who's connected to who, basically. Okay, that's very cool. I guess I have a more basic question. This transition from doing this type of work as a postdoc to doing it in your own independent lab. What are some of the What are some of like the interesting learning curves that you have to go through as you're building your lab and you're starting to do experiments in the lab? Like, what were you prepared for? What was surprising? What were some of the challenges? I guess from the perspective of somebody late in their postdoc or something, what could they be anticipating if they're thinking about starting up their own lab?
Yeah, I think it's been a lot of fun. Let's say that first. There have been a number of challenges. I think everything goes slower than you think it's going to. Every experiment takes longer. Getting everything set up takes longer, and you have to. You wind up having to learn how to make sure you know how to do everything yourself, right? You come out of a postdoc and maybe how to do 90% of the stuff you need to do, and there's the other 10% you got to figure out. So that has been a little bit of a challenge, but I think really, sort of just jumping in and trying to just do it all.
Yeah, because I remember, our time together in the Fitzpatrick lab. You were, obviously doing experiments all the time, doing this sort of thing. You're sort of trained to get your hands on everything. Is it similar when you're starting up the lab? Are you doing a lot of the experiments yourself before you have like grad students and postdocs on hand? I mean, what's the kind of... Is it kind of just like having your own version of what you had going on at MPFI? I guess?
I mean, in some ways it is that you at least initially, at least for my lab, I started out with one technician, and so it was sort of like, okay, let's see what we can start doing. I had one graduate student join relatively early, and so that sort of helped. This graduate student's been really great and sort of helped me collect a whole bunch of preliminary data for every project that we've done. Some of which are still in sort of preliminary data phase, some of which have now become published papers, and sort of getting that, taking all those ideas you have and sort of translating them into actual bits of data that you can then turn into grants. So getting that crank moving.
Mm-hmm.
It took a little bit longer than I anticipated, but I think we were able to be fairly successful doing it.
And so, what does the lab look like now? You're probably about, I would say, 4 or 5 years into running things. So what is the lab looking like nowadays? Like, how many people do you have working on projects in the lab right now?
Yeah, so right now we have 2 graduate students, one fifth, sixth year, one third year, I think, and then one postdoc, and 2 technicians, and then myself.
I saw on your website that your postdoc was building a bike, it looked like.
Yes.
It's pretty cool.
Yeah. Yes, our postdocs, we strive for sort of work-life balance. I think it's important. I think it's important for everybody. And so
I
yes.
feel like it's a good if you've got a postdoc who in their spare time is building complex pieces of machinery, it's probably a good sign of like what they can do with their hands. I don't know. So one question I had for you was, we're back at SFN for the first time in a while. We had this pandemic hit. How much of the startup and the building of your lab were you able to get done prior to the pandemic hitting? What kind of impact did that have on you as like an early stage PI basically?
Yeah, it was definitely a struggle. I think I got a little bit lucky in that I had one of my students, one of my postdocs who were in the lab prior to the pandemic, and we had sort of done most of the training, and they had techniques, they had projects that were working. And so those two people were able to, by and large, continue with their projects. Some slow down, but weren't impacted terribly. I really felt for the people who tried to join the lab or who did join the lab during the pandemic, that it was a really challenging time to try to teach anyone anything. You're trying to remote teach some surgical technique or something like that. And it was really challenging. I think there's also a sense of this sort of institutional knowledge or this sort of collective that happens when everyone's in the same room and talking to each other that we've missed for a couple of years. And I think that that does have an impact, not being around, not being in the same place and being easy to talk to each other.
One thing that I'm hearing a lot about these days is, shortage of postdocs, shortage of people that are sort of in the life science pipeline. Is that something that people in the sort of like junior, assistant professor level, position are like more vulnerable, you think, to? Or, and also, are you hiring right now? This is probably
yes.
a good advertisement for the Smith Lab at University of Minnesota. Who might be listening to that? We got a lot of grad students and postdocs listening to this podcast. Like, who might be, thinking about like, maybe I should check out the website, see what's going on in the Smith Lab.
No, we definitely are hiring. I think we have a couple of really exciting projects. We've done a really nice job of getting some nice funding, so we're well positioned for a lot of things that are really exciting. It certainly is a challenge. I think that the pandemic definitely caused people to sort of rethink what they're doing and how they want to do it and decide how they want to make that work. I think junior labs are perhaps more vulnerable in the sense that they don't have this international reputation to attract students. But I think there's also something to be said for working for junior labs. There's a little bit of maybe freedom or opportunity there that's different. But I think what I've always felt is making sure that you find a good mentor, someone who you can interact with well, who is a good supportive environment. I think I don't want to tell any stories, but I'm sure we can all imagine there are people we might have interacted with that might not be. I was very fortunate to work with David, who was a great mentor. And so that's sort of what we aim to do in my lab as well.
Yeah, I mean, and you must be doing something right because you've, pretty quickly put together a good group of grad students and postdocs. What is kind of your approach to mentorship? Did you have a lot of experience with that in your postdoc days? I mean, how did you figure out that aspect of the job? Because it's such a big part of the job, I imagine.
Yeah, I think it's. I'm still figuring it out. It's still a little bit of trial and error. Try to just sort of start from the basics of be a good person and have respect for the people in the lab. I think everyone wants to work hard. Everyone's interested in the science, and it's just try to make sure you position people for what they want. Some of my students are very interested in academic tracks, and so we can try to talk about how to position them for that. Other of my students are more interested in industry positions, and so we can think about they're not necessarily, maybe you don't do the exact same project, right? Maybe your goals for a project are different if you're aiming for an industry position where maybe you don't want to have this 10-year project that leads to a Nature paper. Maybe you want something that's a little bit more tractable and faster that gets you positioned for the job that you want.
Yeah, I think that's a really important thing to consider is like what's the style of like of the work that you want the people in your lab doing, and it might not be the same from one person to another.
No, and I don't think it is. Not everyone's project should be the same, even though we're all interested in the same science, that people have different goals, and you want to build a project that works for the lab, but also works for the person. It doesn't seem much fun to try to shove someone to do a project that they're not excited about.
Yeah. I mean, maybe we could take a minute to sort of plug what's special about University of Minnesota at the moment, like especially if, when you were brought on, it seems like there was a lot of focus on hiring people in the areas of, optical approaches for understanding the brain. What is that sort of scene like? Is that kind of a big component of what's going on in your department right now? What are some of the broader goals of what's going on? Is, there's a lot of, different identities for a lot of departments around the country in the U.S., and it seems like this is a big part of what's going on at University of Minnesota.
It absolutely is. So I was brought on as part of this optical imaging group, and it was about a $30 million initiative to bring in about 6 or 7 faculty. Now we've successfully brought on those faculty over the past 5 years, and so we have a group of people that are imaging, doing optical approaches, 2-photon imaging, wide field imaging, 3-photon imaging, doing some spine resolution imaging. We recently hired a couple people who are making some voltage sensors. Then we have some computational modelers in the group. So it's a really exciting group of all fairly new faculty that are all really collaborative and working together to sort of leverage the ability of optical approaches to collect these highly dense data sets and then make sense of them. So that's really exciting. In addition to this optical imaging group, there's also a parallel $30 million initiative to bring in a lot of addiction faculty. So the department, I think, over the last 5 years has hired, I want to say 10 to 15 junior faculty. So it's really a growth phase. It's a really exciting time to be there.
That's really cool. I personally have a lot of fondness for University of Minnesota because my stepfather was in a lab studying brain cancer at University of Minnesota. I think maybe at the veterinary school. I'm not exactly sure where it was. He took me around the lab, and that actually was like a big part of me deciding I wanted to like go to graduate school. Just seeing like how like all the different everything that went into a single lab's research like was really inspiring. So like shout out to University of Minnesota for
it's a really good environment,
being a really cool place to hang out. And how is life in Minnesota? Like you're from, where are you from originally?
I grew up in Indiana,
Indiana, like me,
and then spent time in Boston and then Florida.
Go Colts.
Yeah, yes.
That's right. And so Minnesota's new for you then?
Yeah, Minnesota's new. It's good. It's got some nice things. That I'm not going to lie to anyone. The weather can get a little cold in the winter, but that just means you get a better coat and you go outside and you have fun anyway.
Yeah, snowmobiles.
Snowmobiles, ice fishing, whatever else people want to do.
That's really cool. Well, all the best to you. It's really great running into you again here at SFN. Thanks for stopping by, and best of luck with all your research coming up in the next few years.
Thanks a lot. This was a lot of fun.
Yeah, appreciate it, Gordon. One of the great things about SFN is you're always running into old friends that you've met, from grad school and postdoc years, and this is no exception. I'm joined today by Dr. Wanchan Oh. He's an assistant professor in the Department of Pharmacology at the University of Colorado at the Anschutz campus. That's in Denver, right?
Yep, that's physically in Aurora.
In Aurora,
Yeah,
okay.
that is medical school.
The medical
only.
school.
Yep.
Okay, great. Well, it's great to see you, Wanchan.
It's
been
very nice to see you.
multiple years since I've seen you last, when you were a postdoc at MPFI. So you have a really interesting trajectory in your research career. So as a PhD student in Karen Zito's lab, you were studying synaptic plasticity of excitatory synapses. Yep. That was right. And then as a postdoc, you were studying inhibitory synapses.
Yes.
And now as an assistant professor, you're studying both. So can you talk a little bit about that progression? What were some of the main choices you were making in your career trajectory? Why did you switch to inhibitory stuff in your postdoc years? And what's different about what you're doing now as a associate professor, assistant professor?
That's great. Okay, thank you again for inviting me.
Yeah.
And such a great opportunities to meet you in person and to share some of our work.
Yeah.
And as kind of independent researcher at University of Colorado Anschutz.
Yeah.
So as Joe mentioned, my PhD work was all about excitatory synapses. So specifically, we look into how dendritic spines, where most of excitatory synapses occur, in hippocampus, of course, including in the cortex, how those spines are eliminated.
So what is the process by which dendritic spines are? A lot of times when we're talking about synaptic plasticity and memory formation, you talk about spines getting bigger through LTP. So you're talking about LTD,
yes,
is that
exactly.
right? Long-term depression?
So by the time I joined Karen Zito's lab, that is in 2009, and there's a lot of studies showing how spines grow and how spines become stronger by LTP, long-term potentiation mechanisms. But we didn't have much information about how spines are eliminated or how spines are shrinking in activity-dependent manner. Of course, in disease model, there are many disease models showing spine elimination or spine density is decreased. But we didn't know the mechanistic part of spine elimination or spine shrinkage and LTD, long-term depressions. So in Karen's lab, we had two-photon microscopy combined with the two-photon uncaging of glutamate. So we were able to mimic glutamatergic activity by using focal release of glutamate. So I was able to identify long-term synaptic release of glutamate is able to induce synapse-specific spine shrinkage and long-term depressions.
So what, like, in terms of the overall theory of how that's involved in like the function of the brain, why is it important to get rid of synapses?
So that is our kind of sort of my second project in Karen's lab was more relevant to that question because during the early development, there is a tremendous amount of new spine formations. And we still don't know yet if those every single spine formed during maybe in humans for the first 5 years or 3 years, and in mice for the first 2 weeks. And we don't know if those spines are becoming functional. And even if they are not functional, then there is the period of decline in synapse pruning. And maybe some of the spines really engaged into functional circuit. They survived. And some of them, for example, if the spine receives less activity, glutamatergic activity, then they may eliminate. So it's not just, we study single spine levels of spine elimination back in Karen's lab, but no, yeah, at circuit level, we don't know yet exactly how those spine elimination really involves in the circuit function. But spine elimination can be linked to circuit formation and circuit maintenance.
You know, I guess what you're kind of saying is that during development or during, you know, different phases of, you know, the brain's life cycle, there's going to be turnover and formation of spines for the purposes of regulating the normal functioning of the circuit. So you can't just accumulate spines forever. You also have to get rid of unnecessary spines. Okay, that's very cool.
Yeah, that is also during the PhD, we also look at when we activated the multiple spines by uncaging of glutamate, inducing long-term potentiation. Then we look at nearby unstimulated spines. Then we observed those spines that are not activated by glutamate are decreased in size and decreased in function.
So is there some amount of like regulating, like maybe there's just sort of a kind of, you know, limited number of resources for supporting a certain number of spines, and like, you know, the ones that are sort of like, you know, fire together, wire together spines are being supported, and then everything else is kind of like falling off. Is that the idea?
And also supporting that, or maybe against of that idea, because what we did was we stimulated multiple spines by uncaging of glutamate. But then our idea was if that is really the source of maybe PhD95, then if we block on downstream pathway, then if the source is the key, then we may not see spine shrinkage. Or we may see spine shrinkage even if the rest of spines increase. So we block calcineurin, for example, but then we were still able to see spine enlargement of stimulated spines, but spine shrinkage was abolished.
Interesting.
So it's not really the limiting factor. Is not the source.
Yeah.
I think we think it's really downstream pathways and between excitatory, inhibitory, stimulated spines and unstimulated spines. Yeah.
So, what sort of inspired you to, like in your postdoc work, switch from doing these experiments of excitatory synapses and uncaged glutamate and that sort of thing to looking at inhibitory synapses? And how did you do these experiments where you were studying inhibitory synapses?
So after my PhD on excitatory synapses, and I thought it's LTP, LTD, then wow, this is really a good regulation of excitatory synapses. But when we kind of zoom out a little bit more, then there is a different types of synapses, inhibitory synapses. And although inhibitory synapse is mainly formed on dendrite, we kind of still don't know how inhibitory synapse is formed initially. And what we thought with Hyungbae Kwon, now he is in Johns Hopkins. And back in 2014, when I joined the lab, we thought, let's look at how inhibitory synapses are formed initially. And then the plastics part is another source of research or another scope of research. But we were really interested in how inhibitory synapses are formed during early development. Then,
What was your first guess? Like, what were the first set of experiments that you were doing?
so because I was doing a lot of uncaging of glutamate, and then nowadays caged compound of GABA, which is inhibitory neurotransmitter, the major neurotransmitter. Caged GABA is commercially available, but back in 2014, we had one set of caged compound, but it was really sensitive to one wavelength of laser.
Yeah.
But luckily, Hyungbae had a caged compound that we can use and we can tune to laser to certain wavelengths. So I was able to easily release GABA using two-photon microscopy and two-photon caging.
So did you guys have to make caged GABA yourself in those early days? Like if it wasn't commercially available, or
I think Hyungbae, we had this. It's not we didn't make it, but we asked a company to make caged compound GABA.
Okay. So it's kind of like before there's like a out of the box like AAV for whatever construct, you can like get a company to build it for you and then sort of custom make it. And now it's more conventionally available. Is that right?
Yes.
Okay, that's cool. So you were the kind of there like at the early stages when it wasn't like a conventional thing to do.
Yes.
You're uncaging GABA. What were some of the things you were finding when you did that?
Because before we started the experiment, first we just look at, because as I said, inhibitory synapses are not formed on structure like spine. So we can't visualize where inhibitory synapses are located. So we labeled inhibitory synapses by Jeffrin.
Right. I remember this. Like the Jeffrin synapse was like the main crux of that big paper that came out when you're at SFN. So you would find the Jeffrin synapse, or could you basically create Jeffrin synapses by uncaging GABA?
Yes. So that is the whole story of my postdoc research. So we first look at how inhibitory synapses are formed during development, because we didn't know if inhibitory synapses are formed in mice, maybe 5 days after birth, or maybe 15 days after. So we had to narrow down when inhibitory synapses are really actively formed. So we just took imaging from corica slices expressing TD tomato for cell fill and Jeffrin GFP or kill Jeffrin to identify where inhibitory synapses are formed. And then interestingly, we observed the tremendous increase in inhibitory synapse density between like P5 in mice, P5 to maybe P10-ish.
So like a week after they're born, they start to form inhibitory synapses. They're not born with these to begin with.
I think still inhibitory synapses are formed even in adult, but maybe the formation of inhibitory synapses are probably under different mechanisms. Maybe during the plasticity or during learning and memory. Still inhibitory synapses are formed, and again, excitatory synapses can be formed as well, but we really were interested in how initially inhibitory synapses are formed during that early development when the circuit is starting to form.
That's really cool. And so, where did that research take you? Like towards the end of your postdoc stuff. So, you were able to identify like a period in the early life where these things were forming really rapidly.
And then uncaging of GABA now is kicks in.
Yeah.
So because I was well trained by Karen about uncaging of glutamate. So basically for me, I just
it's kind of the same thing, right?
Same thing.
Yeah.
But I moved from hippocampus in Karen's lab to cortex in Hyungbae's lab, and also we combined in Hyungbae's lab ex vivo slice imaging and electrophysiology combined with uncaging of GABA, and also we were able to do uncaging of GABA in vivo mice.
Very cool.
Yeah, and then basically our hypothesis was, okay, this is the period where GABA is inhibitory synapses are formed a lot. And we used GABA on caging, and we released GABA onto dendrite. So
Yeah.
in a mice, in mice where GABA release is significantly decreased, then people observed the decreased inhibitory synapse density. So that kind of indicates inhibitory synapse formation is GABA dependent or inhibitory synapse dependent or inhibitory synaptic activity dependent. That's why we released GABA on dendrite, and then we were able to form new Jeffrin cluster along dendrite.
So I guess in terms of these different experimental approaches, doing it in vivo versus doing it in slice, what are the different types of things you can solve by doing those different approaches? What do you learn from doing it in vivo that you couldn't learn from doing it in slices, for instance?
For example, in slices, what we found was when we release GABA locally by GABA on caging, we were able to form inhibitory synapses on dendrite. But here, the age is very critical because when we observed that there is a huge number of increase in inhibitory synapse formation around P7, P8. But then after maybe P12, P13, when I released GABA onto dendrite, then success rate of inhibitory synapse formation was significantly decreased. Then there's something is going on in that
window. Yeah.
And then, long story short, we found inhibitory neurotransmitter GABA acts as excitatory neurotransmitter before P8 in mice because chloride ion is so much higher
That's right, yeah.
inside of the cell. So when we release GABA onto dendrite, then chloride ion is leaving out of the cell. Then the dendrite becomes depolarized.
Right.
And that's why we observed inhibitory synapse formation together with excitatory synapse formation by GABA on caging at that age.
That's so cool.
And then here's in vivo part is critical because we make slice culture or occur slices, but anything could happen because this is ex vivo. So we really want to verify our age dependent and GABA dependent inhibitory and excitatory synapse formation in that age
In
is
the whole
really,
brain, yeah.
it's true. So we made a small cranial window at P8 of a pop. Then we performed in vivo GABA on caging, and we observed both excitatory and inhibitory synapses are formed by local release of GABA. So that is, in our case, how in vivo study is really critical because we really want to verify this is not the artifact
Of something going on in the slice.
Exactly.
That's so
exactly.
cool. And then if you wait longer, the GABA will just form inhibitory synapses?
No, then nothing happened.
Nothing happens.
Nothing happened. That's why we think it's really calcium dependent. And because we observe in early stage, it's depolarization by GABA because the chloride ion is leaving, and then voltage gated calcium channels are activated. Then calcium influx, and we don't know yet downstream pathways, but it's calcium dependent excitatory and inhibitory synaptogenesis by GABA. And then when we perform the same experiment using mature slices, then nothing happens. Although, as I said, inhibitory synapses I think still are formed and eliminated in adults. That's why I speculate those inhibitory synapse formation and elimination, that plasticity may involve different pathways or different mechanisms.
Interesting.
Yeah.
That's really interesting. So it sounds like at the end of your postdoc, there's a lot of questions that are sort of excitatory and inhibitory,
Exactly.
and it makes perfect sense that when you started your lab in Colorado, that would be like kind of your main interest. So what's the new spin on these experiments that you went after when you first started up the lab? So what year did you start the lab, by the way?
May 1st, 2018.
2018. So like 2 years before the pandemic hit.
Exactly.
You had enough time to sort of build some equipment and sort of thing, get some experiments going.
If I knew we would have pandemic, I could do better. But yes,
Yeah.
I had about 2 years before everything was shut down.
So what was your first priority experimentally when you started the new lab in 2018? Like what was the first set of experiments you wanted to do?
So what I did was, again, my background in excitatory synaptic plasticity in Karen Zito's lab, and inhibitory synaptic plasticity and formation in Hyungbae Gon's lab. So then I thought, maybe I can do both. especially how excitatory and inhibitory synapses are talk to each other, for example. Because again, over the last decades, a lot of studies focused on how excitatory activity regulates excitatory synapses, and inhibitory activity, which is GABAergic activity, regulates inhibitory synapses. But we still don't know yet how those excitatory and inhibitory synapses crosstalk and influence each other. Then one really good spot for this research question was double spines. So double spines are excitatory synapses, but receive inhibitory input as well. So it is really morphologically just a spine.
A normal spine.
Yeah, normal spine, but it has Jeffrin, and it also expresses GABA A receptors, and we also think it expresses GABA B receptors. So those double spines are kind of the spot where I thought, okay, this is really good research area because within that such a small spine, it receives both excitatory and inhibitory activity.
So like maybe I don't know if this is like an easy statistic to name, but like what percentage of
Yep.
dendritic spines are, purely glutamatergic spines versus double spines versus whatever?
We still can't really say what percent of spines are excitatory because our way to label double spines are spine with a Jeffrin. So maybe it is underestimated, but that's because Jeffrin is not going to be labeled by our Jeffrin intrabody technique.
Yeah.
However, we observed in hippocampus when we look at distal dendrite, then roughly 20% of dendritic spines are Jeffrin positive. So those are double spines.
So it's like maybe 1 in 5 spines. It's not like
Exactly.
an overabundance of these things. They're kind of a specialized class.
Yes,
That's
it
very
is.
cool.
Yeah, and interestingly, this is 1 out of 5 spine, double spines. But in terms of inhibitory transmissions, about 30% of inhibitory inputs arrive in double spines. So it's quite big number. It's
Yeah.
really small number of spines, but it's really substantial. Of spines are double spines.
Yeah.
Because in the lab, when I started the lab, we set up our two-photon microscopy using 3 two-photon lasers. So one laser for imaging, and second laser for uncaging of glutamate, and third laser for uncaging of GABA. So which means we can control both glutamatergic activity and GABAergic activity by releasing GABA and glutamate almost simultaneously. So we can study how inhibitory activity regulates excitatory synapses, or vice versa.
And I think you said before we sat down to talk that this is going to be published in Neuron tomorrow.
Tomorrow will be out online.
That's awesome. So what is the sort of main punch of the paper, the main result?
So double spines have been observed often, and that more than 2 great studies from Ellie Nedivi's lab at MIT using triple color two-photon imaging in vivo in visual cortex. They observed double spines are incredibly stable. So when they performed in vivo imaging every day or every other day, they found overall about 80% of spines are dynamic. And then if spines are lacking PHC95, which is maybe immature spines, those immature spines are much more dynamic. But when they focused on double spines with PHC95 and Jeffrin positive, then more than 95%, I think, are stable. So they don't undergo
Wow.
plasticity. And then from there, we thought, oh, maybe double spines are resist structural plasticity, such as LTP, which is well-known important synaptic mechanisms underlining learning and memory and synaptic strengthening. So we used first uncaging of glutamate to make spines undergo LTP on single spines. And then many people have shown high frequency uncaging of glutamate can induce LTP and structural long-term potentiations, which was also true for our hands. So we made the spines bigger in hippocampus, especially CA1 pyramidal neuron. And then we used those identified, verified uncaging protocol onto double spines. then see if double spines are undergoing LTP or they are really resist. They don't undergo. Then surprisingly, no, they stay there. They don't undergo LTP.
Wow. That's
And
really
then
cool.
was very interesting. But then, as many of you know, NMDA receptor is the main component underlying synaptic plasticity, including LTP. Then we thought, oh, maybe NMDA receptor is impaired or something is going on there.
Mhm.
Then we used same uncaging protocol, uncaging method, but we combined now electrophysiology and we measured NMDA receptor current at single and double spines. Then we observed NMDA receptor current was significantly impaired at double spines. And what's very exciting, and we further explore what's the mechanisms, because the key difference between single and double spines are whether they have inhibitory input or not. So now we used two-color uncaging. So uncaging of glutamate, uncaging of GABA.
You have these double spines. They're sort of NMDA current defective. They're not undergoing crazy structural plasticity with like rapid uncaging of glutamate or whatever. Do you know anything about when they emerge developmentally? Because it seems like maybe they're like this kind of like stable force for maintaining like the structure of the network or something like that. Is there anything that you've learned so far about that, or is that an open question?
Yep, that is a super interesting question. We also try to address that question because plasticity part was, okay, double spines are impaired in terms of NMDA receptor function. So maybe that's why they don't undergo LTP. And we found it is GABA B receptor dependent pathway at double spines. Then another question is then when those double spines are formed? Because in this field, synaptic plasticity field, I think the conventional idea of double spine formation was spine formation was when single spine receive a lot of glutamatergic activity, and when they undergo too much excitation at single spine level, then somehow single spines recruit inhibitory components, then becomes double spines. That is what many people thought. That means if we block excitatory transmission or excitatory glutamate release from CA1 area, then CA3 area, sorry, then CA1 pyramidal neuron will receive less glutamate.
So then they would, under that hypothesis, they'd have less of these double
Exactly.
spines.
Then our hypothesis was blockade of glutamate will lead to maybe no double spines or less number of double spines. Then this is a collaboration with Matt Kennedy in our department. So Matt Kennedy designed and made a virus so we can specifically abolish glutamate release from CA1 neurons. And then we did recording imaging from CA. Oh my God, can
block
you?
CA3, yeah.
So we block on glutamate release from CA3 area, and then we look at CA1. And if our hypothesis was correct, then we would see less double spines or maybe no double spines. However, even if we block glutamate release from CA3, verified by electrophysiology and mGrasp, nothing happened.
That's
Still,
crazy.
double spines are there. They are there.
That's so interesting.
It is really excitatory activity independent.
Like what blows my mind is like, textbook neuroscience has been talking about LTP and LTD and, excitatory inhibitory inputs to cells for decades. And we still are finding new information about this stuff. It's like a never-ending pile of questions about how these things are formed and what their role is. So like it's must be such a rewarding field to work in because you're just surrounded by mysteries all the time. And so that's really cool. Another question I wanted to ask you is like, you mentioned you started in 2018 with your new lab. The pandemic hit in 2020. What was the impact of that on your, like, now we're back at SFN. Everything seems like more or less back to normal. Like, what were the last few years like for the lab in terms of like keeping things running and stuff? Obviously you got this paper coming out, so things are going pretty well.
So this double spine is one of our projects, and we, as a lab, we mainly study neuromodulation. For example, how serotonin or how dopamine or even how nicotine
Mm-hmm.
modulate excitatory and inhibitory synaptic plasticity and formations. That is because two-color uncaging, which we use a lot mainly in the lab, for this double spine experiment or research, we can control glutamate and GABA. But there are caged compounds of neuromodulators,
Like
such as
serotonin and stuff.
serotonin, for example. Then we can release the serotonin and we can release glutamate onto dendrite, onto spines, on different ages of animals. Then we can study how those neuromodulators regulate synapse development. So that was our main focus in my first 2 years, and I had really good first rotation students, Roberto Ogelman, and who decided to join the lab, luckily, and we got some good amount of data, and we submitted my first R01. Then with the first submission, I got R01.
That's
So
amazing.
that is before pandemic hit.
Oh my gosh,
So
that's so
I
lucky.
submitted early 2020,
Yeah.
and then so luckily, very fortunately, pandemic didn't impact our research a lot. Although it's like for everyone, there's a shutdown period, so we couldn't do anything. However, at least with Roberto and me, we were able to discuss a lot. And Roberto prepared his comprehensive exam, and he prepared for his F31 grant using some of our data that we collected before pandemic. So he's doing great.
That's good.
And we have found some intriguing findings of how serotonin modulates dendritic spines in prefrontal cortex.
Yeah, so like, I mean, that's really great to hear because I feel like in neuroscience in general, especially when there's questions about when the grant funding is going to come in, sort of that sort of thing, it seems like the labs that are going to be the most vulnerable are the ones who are just starting up. Like, you know, the ones who are just sort of like getting all their stuff built and getting the experiments going and recruiting grad students and recruiting postdocs. So it's really great to hear that you were basically able to sort of like get through and, you know, make everything work because, it's really good. And how are things now? So I guess this is the part that I'm really excited about. Are you hiring right now? Are you looking for grad students, postdocs, anything like that? Like anybody who's listening to this podcast who's really interested in, synaptic plasticity and inhibitory and excitatory and neuromodulation of synapses, you know, what kind of students should be applying or sending you emails right now, basically?
Oh, that's great. Thank you for these opportunities. So yes, we are hiring at any levels of scientists who is really dedicated and self-motivated because it's a very sophisticated experiment.
Yeah.
We have to have good slices and good hands on electrophysiology, and we have to control 3 lasers simultaneously. But my research background, before I joined the Karen's lab, I had 0 background in electrophysiology, and I had 0 background in 2-photo microscopy. But I survived, and I went through well, and that means anyone can do that.
That's great.
With good training, of course.
Yeah. So I mean, and I you know, knowing you from like the years that we both worked at MPFI together, I feel like, you would probably be a really great person to come in and learn it because you're very warm and, genuine person who's really invested in people and like really cares about the science and stuff. So I think anybody who feels daunted by the complexity of the experiments, I bet it would be a really good, my own personal endorsement would be probably a really good environment to learn all those techniques. And so look him up. He's his lab website, I'm sure will have all the details of how to get in touch with him.
Exactly.
So Wanchan, thank you so much for sitting down and talking to us today. This is really great. It's really great catching up with you and hearing about your research trajectory and how everything's going these last couple of years.
Great.
Have a great rest of the meeting.
Thank you so much.
Take care.
It's very nice to see you.
Great to see you. Okay, so we are back. We're on the floor of SFN 2022, and I've said this before, and I'll say it again. But like one of the best things about coming to SFN is like running into friends from past and present. And I'm joined right now by my good colleague from Florida Atlantic University, who we have a really strong partnership with at MPFI, Dr. Bethany Stanhope. She's the Executive Director of Academic Operations at FAU's Jupiter campus. Bethany, thank you for sitting down on the podcast.
Yeah, thanks so much for having me today, Joe.
So we were talking yesterday when I ran into you at the MPFI booth, and you mentioned that this was the first time you've been to SFN. So I was like, okay, so we got this podcast up. I got to talk to Bethany about her initial impression of coming to SFN for the first time. So you're no stranger to scientific meetings. So can you talk a little bit about what you did for your PhD work and your postdoc work?
Yeah, so I started my kind of journey in science looking at evolutionary traits, and in particular, I was looking at the evolution of pigmentation. And so I started during my PhD looking for genetic regulators of pigmentation and coloration development. And that brought me to a very cool study system called the blind Mexican cavefish, which you might have seen pictures of. You know, it's this little albino-looking fish that evolved over a couple million years in central Mexico. And so the cool thing about this fish is it has an extant, so a living surface-dwelling morph, which are completely interfertile. And so you could actually cross them together and do genetic mapping and pedigrees to actually link genes to specific traits of interest. And so that was kind of my first journey into genetics and really looking for regulators of specific traits. But ultimately, kind of what I found during my PhD program was I really loved linking genes to phenotypes, but admittedly, the world of pigmentation stuff has been a little bit overdone, a little bit exhausted. So I finally was like, well, I need a challenge. So that brought me to working with Alex Keene at Florida Atlantic University back in 2015, where I continued my work in cavefish and actually adapting transgenics, CRISPR, to cavefish to link genes of sleep disorders in cavefish. And then I also overlaid that with kind of expanding my skill set and also learning all the tools in Drosophila so I could do large genetic screens and again look for genetic regulators of sleep.
Okay, so that was kind of where you shifted into the neuroscience world a bit, is like in your postdoc work.
Right.
So backing up to these blind Mexican cavefish, because I don't think we've talked about them on this podcast before, and I think they're super fascinating. So what you're saying is like there's different versions of this species of fish that live in specific caves in Mexico. Like you could actually go to a specific cave and like find a couple of these fish here and there, and they have differences in their gene expression patterns, but they can interbreed with each other. And then there's a version of this fish that has complete eyesight, isn't blind from like, you know, evolving in the dark and that sort of thing. Is that the basic idea?
Yeah, absolutely. So yeah, the system is pretty amazing. And so it's kind of a natural, almost, I like to think it's similar to like humans of different lineages. So several of these cavefish populations actually got isolated about 2 to 3 million years ago in these about 30 different caves in central Mexico. And they oftentimes independently converged on a similar suite of traits. And so the most obvious ones are the loss of eyes and loss of pigmentation. But what people didn't realize at first is the whole suite of changes that kind of come along with the changes of behavior. And so as you can envision living in a completely dark environment, trying to figure out how you find food, you have expansion of all these other traits, you know, like sensing the walls and being able to find food, and they're much more sensitive about these things. But we actually started looking at their brains in the last few years and saw that there were these gross kind of morphological changes. If you think about your brain, if you actually pluck out an eye, you actually have a lot of collateral space to kind of expand other systems and other regions of your brain. And as well, they also, in particular, we studied the changes in their sleep patterns. Because if you imagine, you don't necessarily want to be asleep if your only food washes into the cave. So they also evolved to become a semi-like insomniac. So they only sleep about an hour or two a night compared to their surface-dwelling countermorph. Yet they're still healthy. So we need to figure out that magical concoction of the cavefish anti-sleep pill that still keeps them alive and well.
So I guess, you know, part of this is that they don't have the normal regulators of sleep that somebody who would be very dependent on like fluctuations in light would have, basically. And also this thing that you're mentioning about your food source could wash into the cave at any time. You need to be sensitive to that.
Right.
It's not really tied to like the day-night cycle or whatever. So
Yeah, absolutely.
so that's why they're really interesting for sleep, I guess. That's really cool. So now you're in this new role where you're like, when I think about all the people doing all the different stuff at Florida Atlantic University, where we have a lot of like academic partnerships, there's one person at the center that's of everything, and that's you. So how did you take on this new role after your postdoc work, where you're basically, from my point of view, like the go-to person when I have a question about anything going on at the university, basically?
Yeah, I think what I really liked about changing directions was kind of the unlimited possibilities that exist on this amazing Tri-Institute campus, where we're neighbors with Max Planck, we're also neighbors with UF Scripps, and really we're kind of all both research yet academic institutions. And so I was kind of presented the opportunity to figure out ways and figure out creative possibilities to how to link research with the training environment and academic environment. And I think in order to do that, since this was kind of a new kind of area of really expansion for like FAU and Jupiter, is I found that the best way is just to get your hands dirty and like immerse yourself in every single group on campus, really to get to know what's going on, because it's really hard to create these partnerships and develop programs if you don't really know who is at the base of like the program and who's really working with the undergraduate researchers on campus. And so it really was critical for me to immerse myself in all those groups to be able to do my job the best, to kind of figure out who the players were in these different groups.
What were some of the things that when you stepped away from doing like full-time postdoc research stuff to doing your current job, what were the stuff that you hit the ground running, like fully prepared for, and what were some of the biggest learning curves you had in your new job?
Yeah, that's a tough question. So, I mean, the easy part was kind of expansion of these course-based undergraduate research experiences, which we call CURES for short. And that's really like taking a small group of undergrads and having them do authentic, original research in a classroom setting. It was a little challenging figuring out the pace at which to do a lot of the research work, but really it ultimately felt to me like a continuation of my postdoc. It was really a lot of mentoring, working with like small groups of students, and really it just becomes kind of an everyday. You meet once a week at your session, you kind of meet up with each group, figuring out where they're at in their projects and giving appropriate feedback. So that kind of felt like an extension of what I had been doing on the postdoc, just in a slightly adjusted way for kind of the more undergrad learning mindset. I think the more challenging part was kind of getting to kind of get a sense of some of the administrative components of working at a university, which I think when you're a scientist, you're just completely, immune to like what's going on in the above world. And so that was like bigger challenging of trying to figure out who do you talk to for certain things. If we want to get $100,000 in funding for, a particular project or initiative, of figuring how we can actually make that happen. Starting to think about things more programmatically was a challenge, and something I had to like learn along the way.
Yeah, I feel kind of similarly. One of the things that strikes me is like, I'm curious to see your impression of this, but like it seems like one of the real main areas of priority for, especially at the Jupiter campus, just because that's what I'm most in tune with in terms of what's going on at FAU, but like there's this huge push on undergraduate research experience. Like you mentioned these course-based undergraduate research experiences, but there's all these symposiums. There's the Office of Undergraduate Research and Inquiry. There's all this stuff going on at FAU where the focus is on getting undergrads into lab environments, doing original research and all this stuff. And, can you talk about some of the different types of opportunities students have? It seems like there's a variable range of different styles of research experience people can get, different sources of research experience people can get. If people are looking to FAU as a place to do their college experience, what are the types of things that they could expect to get involved in?
Yeah, I think that's something that we've really diversified ourselves in, and I think we have kind of an opportunity for, whatever time frame, whatever length of time you're dealing with, I really think we do have it set up. So, starting at the basics, we have your traditional, you join a lab, you work with like a mentor type of situation, and that's going quite strong. We have students that are working at um, labs in Jupiter. We have them in Boca, down at Davie, but we also have some of our undergrads and interns and also grad students at Max Planck, and you have Scripps as well.
Hmm.
Um, but going aside from the kind of the traditional mindset, which is a growing challenge as an honors campus in Jupiter, where everyone's trying to do a thesis-based project, you have to come up with creative ways to make sure all undergraduates are getting opportunities. And so what we're doing with that are kind of these course-based undergraduate research experiences is one way that we're doing it, where you can, fully CRISPR some zebrafish, you can do some neurophysiology on crickets. And then we also, if you don't have the opportunity to do it during the year, we also have these summer research experience for undergrads as well. So we have a local one that's open to FAU students to continue and get paid in the summer, because that's a critical thing is, a lot of students don't necessarily have the means to just volunteer for the entire summer to do research. So having paid opportunities is really important. And we also have a funded RU through the National Science Foundation, where we can actually bring in undergraduates from across the country and actually experience research on our campus as well, especially for colleges that aren't big into research. So we try to prioritize, community colleges, small liberal arts colleges as well.
Yeah, and actually at MPFI we've started hosting some of those students for summer research experiences, and that's been really great because one thing that has actually blown my mind is transitioning from maybe not having a super hardcore neuroscience background to jumping into like a research lab where the bread and butter is like really hardcore neuroscience techniques, two photon imaging, electrophysiology, that sort of thing. The students are doing such an amazing job of like hitting the ground running, not being intimidated, like doing really great stuff. What is it you think makes a student like from FAU like so adaptable, so able to fit in basically all these different situations?
Yeah, I think we do a really good job in doing kind of basic training and helping students kind of get the concept of, research, because, as I educate a lot of these students myself, I've always felt that the toughest part of science is actually thinking about it and thinking critically. It's much harder to come up with a strong hypothesis than really just doing some cookie cutter experiments, mixing stuff up in tubes that can be taught, but really thinking critically of like, you get this result, and what does this result mean? You have to think about, did you learn something? And sometimes even a failed experiment still means you learned something, you crossed something off the list, and you have to revise your hypothesis and go in a different direction. And I think what FAU does really well is kind of prepare this research mindset of taking something, a curious question, and putting it into practice of using, experiments and thinking about it and really putting it together as a project is a much bigger challenge than, just, mixing stuff in flasks and.
Yeah. I mean, I think one thing that FAU is doing really well is sort of like guiding students through this whole sort of research arc from like jumping into a project and, forming a hypothesis and doing experiments. And then along the way, while all that's going on, they're getting a lot of professional development. They're getting a lot of like instruction on how do you put your poster together? What's the most effective way to communicate this to an audience? Like there's a real focus, it seems broadly, from the high school level up to the grad student level, where there's this sort of like emphasis on quality and communicating your message and all that stuff that I think is oftentimes left up to like individual mentors, but it seems like programmatically it's sort of built into all these different stages really well at FAU. So I've always thought that was really impressive. So you mentioned a couple like summer research opportunities, the one for FAU students and then the one that students can apply to from elsewhere, from other colleges and universities. Where can people find information about that, and when would applications be going up for the next year?
The application will be going up soon with a deadline in early 2023. And so those resources can be found on the FAU Jupiter website, and especially the one through the NSF is also available through there. And, it's not even just starting to look here, but it really exposes you to opportunities elsewhere. So I also encourage, if it's not a good fit at FAU, or really just to apply to as many programs as you possibly can to be able to have the opportunity to do a summer, research experience. It's something I didn't know existed when I was an undergrad, and really wish I would have had the opportunity to do that, to do, an intensive 10-week mentored research experience. Would have just been amazing.
So getting back to your career trajectory, coming from like the world of evolution and genetics to, doing stuff on, genetic regulators of sleep in blind Mexican cavefish and stuff like that. You mentioned that like a lot of the meetings that you went to early on were like, smaller meetings, like 300 person, like symposiums with, like more intimate settings than SFN. So coming to SFN for the first time, like what is your first impression of this 25,000 person meeting, like halfway, all the way across the country?
Yeah, I'm not gonna lie. When I first walked in and I like opened my eyes and they're like, "What's your badge number? What's your QR code?" I was like, I was like, "Wait, what number am I here?" And I was like, "Oh, I'm like number like 22,000, something like that, amongst all these other individuals." Yeah.
Does it
So
say? Oh my gosh.
yeah, I think your badge
That's funny.
number.
I'm like 10,000 something.
Yeah, no, I was a late register, so I was up there in the numbers, but it was, and being like shuttled through lines and stuff like that, and so it seemed, admittedly, it's slightly overwhelming at first, because you're like, how do you kind of foster like these more intimate interactions? Because ultimately, a meeting is supposed to bring people together, and you don't just want to like exist as a number in a crowd. So how do you set yourself apart, which can always be really challenging, and something that I also encourage like my mentees to do, is you have to actually look up posters, connect with people, attend the socials and stop at booths and actually introduce yourself, because we're all kind of in that together. Of you have to like stand out somehow and not just be a number.
Yeah, I like SFN. You got to get the app, otherwise, like you can't navigate it. Like you need to know when things are happening, where to go, like finding the poster sessions and the symposium stuff. It's so big that like it's a completely different. There's a whole. You could do a whole seminar on how to attend SFN. I feel like.
I think that would be amazing.
Yeah.
But the cool part that I do think is super interesting and different than other more intimate meetings I've been to, is you just get exposure to so many different opportunities all at once, which is really cool, especially if you're an undergraduate or a grad student. You're really trying to figure out like what's your next step. Having all of these pieces all here at once, and not even just the scientists, but also the vendor exhibits is pretty cool too, because you can really learn about the newest technologies. Think about how you can like implement those new technologies into your research, or maybe it's something you're really fascinated in, and at the end of your postdoc, and maybe that's your avenue in science is through biotech or a company or something like that. So that's something that is totally different for me, and I thought that was really cool, was actually talking about some of the development of these tools, with the people that are doing it. So
Is there anything you're looking forward to doing for the rest of the time that you're here? You got a few more days left. Any talks or poster sessions you're trying to get to go see?
yeah, I think for me, I'm a big fan of the poster sessions. As much as I do love talks, and it's a great way to like hear about the most cutting edge stuff. I love FaceTime, with other scientists, and being able to really ask those hardy questions to someone, and give feedback, or just introduce myself and tell them about the programs that we have going on. And so I think that's kind of where I'm set. And I'm looking forward to the MPFI social, so I can
Shout out to the MPFI social, yeah. I'm sure this will be coming out after the MPFI social, so it's not a good advertisement, but maybe we'll do another one next time too.
But yeah, just knowing that, and that's something, if for anyone listening, if they haven't been to SFN, they have all these little sub-social groups, in the evenings and stuff like that. So there are ways to find more intimate experiences.
Yeah, and like get to know people in your field and stuff, like in your subfield.
Exactly.
Yeah.
So I think that's really cool. As something like an added layer that you don't have to just feel overwhelmed by just showing up to a 30,000 person conference, there are these little intimate meetups so you can get to know some individuals, like you said, in your field more one-on-one.
I feel like one thing I really like about SFN is like I'll definitely like block off time to just go randomly sort of search. Like today I was looking at a couple posters because we're going to be interviewing some of the PIs that are running the projects on these things, but then I look at the poster next to it and I see this poster on like how spiders are building spider webs and like what's the sensory motor feedback that they use to like do this. And I was just like, I've been fascinated by spiders my entire life. I've never heard about this. What are the odds I would find a poster that would just like catch my eye, like just by going to another poster? Like the random stuff that pops out at you, I think is always super fun. So I'm hoping I can get a little bit more of that in. Maybe I can find this spider guy and get him on the podcast too.
That'd be pretty cool, especially being in Florida. There's lots of crazy spiders down there, and
yeah,
we've got the banana spiders and the peacock spiders.
Yeah, it's. I feel like the range of stuff that you have at this big meeting is like it never fails to impress. There's always something super cool that you'll leave remembering like for a while. So cool. Well, Dr. Stanhope, thanks so much for sitting down and talking on this podcast. I hope you have a good rest of your first SFN and hopefully not your last SFN.
Yeah, absolutely. Thanks so much for having me, Joe.
Absolutely.